It's filming. You're good.
Okay.
Oh, there you go.
Hey, we're on page 72, section 4.5. We're learning about stuff inside the cell. Now, the endoplasmic reticulum, I talked about it yesterday. It's membrane, phospholipid membrane that sits inside the cell, and it either has ribosomes on it. When it has ribosomes on it, you know what it's called? Rough endoplasmic reticulum. Somebody besides Stephanie, if it doesn't have ribosomes on it. Smooth endoplasmic reticulum. And that's this stuff right here. And one thing that you aren't able to easily see in these pictures is that these pieces of this membrane can break off and travel through the cell. The membrane is always changing and moving a little bit. And so like this piece right here could actually break off and move around and join up somewhere else. So it's kind of a fluid system. And what will happen is, they show it right here. Here's a bunch of ribosomes covering the endoplasmic reticulum, and they've made some substances. This green stuff right here is some kind of protein that has been made by the ribosomes. And you can see that a piece of the endoplasmic reticulum is breaking off here, and it's traveling through the cell. And one of the places that this stuff will go is called the Golgi Apparatus, also known as the Golgi Body. Jessica, you with me up here? This is the Golgi Body. It was named after the scientist that discovered it. His last name was Golgi. And what will happen is this vesicle that's filled with proteins and maybe fats too, that have been made down here in the endoplasmic reticulum, goes into this Golgi body, which is just the membranous sacs stacked on top of one another. Stacks and stacks of membranous sacs. I will not eat green eggs and ham. And they're just kind of stacked up like a stack of pancakes. And these vesicles will kind of move through these membranous sacs, and the material that they bring will be changed in their little journey along. You see, the Golgi body is like a factory. Have you ever heard of an assembly line?
Yeah.
Assembly line works like this. Like, let's say you're putting together a car in an assembly line. The material comes in one side. Are you getting me on film?
No.
The material comes into the factory in one side, and you get metal, and you get tires, and you get leather, and you get glass. And one by one, the stuff is put together. This station puts on the wheels, and this station puts on the doors, and this station puts in the windows, and this station puts in the engine. And by the time it comes to the end, you got a car that's built. It comes out the other end of the factory, right?
Chevy Volt.
Could be a Chevy Volt, if you have that kind of cash. Anyway, it's the same thing happens here. The material comes in this side, and it goes all the way through the Golgi, and when it comes out the other side, it's complete. Yes?
What's the name of that orange thing? Like, is that part of the...
It's called a vesicle.
I mean, like, when it's...
Or
the other part.
When it's come out?
No, like before it does.
Down here?
Yeah.
This part right here is called smooth endoplasmic reticulum, and this part down here is called rough endoplasmic.
So those little like yellow balls are ribosomes?
Those little yellow balls are ribosomes.
So the yellow balls have to go through the smooth and the rough?
Well, the yellow balls make the proteins. Okay?
Yellow balls make
The ribosomes make the proteins, then squirt the proteins into the endoplasmic reticulum, and then the proteins move through here and away.
So it only goes like... I'm sorry, I'm really...
No, you're right.
So it has to go through both.
Mm-hmm.
It could go through both. It doesn't have to go through both. The proteins can go into the smooth endoplasmic reticulum, and in the smooth endoplasmic reticulum, some lipids, some fats are added to it.
And then it goes to the Golgi.
And then it can go to the
So
Golgi.
it
Oh,
could go through the rough and then go to the Golgi.
it could go straight from the rough to the Golgi if it doesn't need to get any fats added to it.
Just one of the endoplasmic reticulums it has to go through, and then the Golgi body.
Yes, yeah. It'll go through the Golgi. Golgi is like a packaging system that puts it into a vesicle. Does that make sense?
Mm-hmm.
Yeah? Good. How about you? You're good? Okay. So now we've got some complete product that eventually can be secreted. Here it shows it being secreted outside the cell. For instance, something like saliva. Saliva is not cells itself. It's secretions of cells. So you have cells in your salivary gland that are creating saliva and packaging it up. And saliva consists of a bunch of enzymes that break down food particles. And so the enzymes are made in the cells and packaged and then secreted outside the cell. And then it goes into your mouth from the salivary gland cells.
What does secreted mean?
Secreted means to be pushed out, to be like what's another word for secreted?
Dispersed.
Dispersed. That's pretty good. To be let go. So, have you ever wondered, have you ever been sneezing a lot and you say, gosh, eventually I'd run out of snot, right? No, you don't. You keep making more. You never run out of snot. The cells will keep making more.
I hate that word.
Snot?
Ugh.
Is it worse than vagina?
Oh my god.
What about penis? I had a teacher that taught this class. It's called Mammalogy, the study of mammals. And we basically, I got my textbook right here. It's kind of a funny story, so I'll go ahead and tell it because it's worth a laugh. It's near the end of the day. You could use a laugh, right? This was our textbook for mammalogy that I took in college. The study of mammals. This was a one-semester class now.
Oh my god.
Look at this textbook. It's freaking crazy. So we start studying, and you come in and we talk about this mammal and then that mammal. We're talking about all the mammals. It's a pretty cool class. But every one of them that we come to, the lady talks about the penis of the male of the mammal. And she says the word penis like that. Penis. That's how she says it. And so the first time, she says it and everybody's like, okay. And then we notice every time she's going into the details of the genitals of the male of that species. And we find out that she wears this necklace to class every day, and it's got a bone on the necklace. And somebody finally asked, what is that on your necklace? And she says it's a baculum, which is a penis bone that that a possum has. A possum is one of the only mammals that has a bone in its penis, an actual bone. And she's wearing one around her neck. Isn't that weird?
Sounds like a weird teacher.
She's really weird. And so we had this student that either came in late or something, or was out and never came to class or something, and came in, and she was saying about some mammal's penis, and she hadn't been to class that much, and she goes, "What's the deal? She's always saying penis." And the girl in front of me turns around and goes, "She likes penises." So I thought it was funny. So anyway, how did we get on that subject?
I don't remember.
No,
it's not.
Now she's like, okay, just to add to this, she's like a 75-year-old woman.
So possums can break their penis?
Yeah, they could.
Oh
yeah,
my god, that's awful.
Very painful.
That is awful.
So let's take a look at some video footage here on the Golgi apparatus. I got some good videos of this stuff. Now here's a Golgi complex right there, and here's a little vesicle. And you'll see the kind of the way it moves. The vesicles move through the Golgi.
Here we see a Golgi apparatus with its cyst face toward us. We also see vesicles and several microtubules. A small vesicle, shown in red, is moving along a microtubule carried by a motor protein. Materials move through the several cisternae of the Golgi apparatus. They are carried in vesicles that bud from one cisterna and fuse with another, or with the plasma membrane.
So that shows pretty well how stuff moves in the cell. You see how this bubble full of material just kind of moves through the Golgi from one thing to another? And it contains, see, this bubble here contains proteins and fats. And within this big membrane here are a bunch of little workers, just like factory workers. Only we don't call them workers, we call them enzymes. They're special proteins that will change the shape of these and add stuff to them or put them together into larger and larger structures. So as this thing moves through, the enzymes in here are acting upon it to change it. Isn't that neat?
Do we have to know cis and trans phase?
No, that's extra stuff. They call the inside face the cis phase and the outside face the trans phase. Is that in your book?
Yes.
Yeah.
Oh, well, maybe you could...
Wait, can you say it again? Cis phase?
The cis phase is the inner part of the Golgi, and the trans phase is the outer part of the Golgi. So that's the cis phase, and there's the trans phase. Whatever's in the book, you have to know. So
Membrane.
if it's not in the book, you don't need to know. See, here's another picture of the Golgi at work. Oh, this is just talking about the Golgi.
Another kind of organelle, the Golgi Bodies, are the cell's packaging centers. Here, proteins are mixed with other chemicals and enclosed in bubble-like containers that then break away and move to the cell membrane, where their contents are released from the cell.
That was awful. Sorry about that. Questions about the Golgi? Now, a cell will have more than one Golgi. A cell might have 20 Golgis or 50 Golgis. They're all over the place.
Is there only one endoplasmic reticulum?
No, there's a bunch of it's all over.
So the things that come out of the Golgis are vesicles.
Are vesicles, yeah, and the things that go into the Golgis are in vesicles. Mm-hmm.
Oh, they're
Vesicles are like little traveling bubbles full of stuff.
What's a ribosome?
A ribosome is
It creates
it's made of protein and RNA, and it creates proteins. It's where the proteins are made to begin with. So these ribosomes make the proteins and squirt it, like this little ribosome here would squirt protein into the space here. And once enough protein got into space, then a bubble would break off and start traveling. Does that make sense? Emery?
I'm a little
Yeah, it's kind of like a game. Yeah.
I'm a little confused. Okay,
Okay.
I thought the little green things in the endoplasmic reticulum, I thought those were proteins.
Those are proteins.
And they're vesicles?
No, they're stored in vesicles.
Okay.
The bubble is the vesicle.
So the bubbles that travel from the endoplasmic reticulum to the Golgi body are the vesicles.
That's correct.
Okay.
Bubbles are vesicles.
What are they called before?
What are the vesicles called before what?
They go to the whatever.
To the Golgi?
No, before that.
Then, well,
Before that.
if
Yeah.
the bubble is down here, it's part of the endoplasmic reticulum. See, the endoplasmic reticulum can break off and form vesicles. A vesicle is just a piece of endoplasmic reticulum that's broken off and is now traveling.
So there's nothing before it gets to that endoplasmic.
No, it is endoplasmic reticulum.
Oh.
It is endoplasmic
It's
reticulum,
just
and then
parts of it, I think.
breaks off. If you break off a piece of the endoplasmic reticulum, you now have a vesicle.
So the endoplasmic reticulum is made of a bunch of vesicles?
Yeah, it's all this material in here. Every little line you see is made of the same stuff. It's made of membrane material, which is a phospholipid bilayer. I talked about this the other day. Let me refresh y'all's memory a little bit. All these little lines that you see, It's all the same stuff. It's this. It's a phospholipid bilayer. That's what membrane is. It's these molecules, these fat molecules that are placed end to end. Remember I showed you a little model of them? Look like this? Did I show you all this, or am I just freaking out?
You did.
This is a phospholipid molecule. Remember a lipid, a fat molecule? If you take one of the fatty acid tails off and hook on a couple of groups, you have one of these things, a phospholipid. And many hundreds of them will make a membrane. And this membrane will form a big bubble. If it forms a big bubble, it's a vesicle. If it forms this big, long, windy thing, it's the endoplasmic reticulum. If it completely surrounds the whole cell, it's the cell membrane. If it's a double layer surrounding the nucleus, it's the nuclear membrane. All the membranes in the whole cell are made of this stuff. So we just have different names for them if they have different shapes. Yes?
Wait, is the Golgi apparatus one of those?
Yes. The Golgi is made of this too. Yes.
Okay.
Okay?
It's stupid.
No.
But what is a membrane?
A membrane is just a surrounding layer.
Of anything?
Of anything. It's a separation. So, for instance, this paper is a membrane surrounding the inside here. Okay? So all the membrane that's within a cell is made up of these molecules. So if I go back, it's a long way. Here we go. If I go back here, that is membrane. That's the nuclear membrane. This is membrane. It's the endoplasmic reticulum is what we call it. This is membrane. It's a vesicle transporting stuff. The Golgi is membrane. It holds enzymes that change stuff. It's all membrane. It's all the same stuff. You can even see here. See how the nuclear membrane here kind of becomes endoplasmic reticulum? They're actually joined together, aren't they? The endoplasmic reticulum is just an extension here of the nuclear membrane.
Can it be separated?
It can be, but it doesn't necessarily have to be. It can be joined.
Okay, so the Golgi Body
That's it.
is made of enzymes? Or enzymes just help it travel through?
It's membrane that surrounds enzymes.
Okay.
And the enzymes change stuff that comes in. It puts things together. It's like a factory. Think you got it? Or is this freaking you out? I got some other ways to look at it that might help you. Let's see some other videos. What?
DNA instructions are chemically copied in the
That's
nucleus.
terrible. We're not looking at that.
The enter-
Sorry. Try this one. That's a good one. Okay. Let's guess what things are. What do you think this is?
Golgi.
That's a Golgi. What do you think this is?
Membrane.
It's the plasma membrane, the outer membrane of the cell.
That's a rough one.
This separates the outside of the cell from the inside of the cell. What do you think this is way up here?
Whoa, yeah.
That's roughy art.
It's roughy art because it's got ribosomes all over it.
Yeah.
Jessica, you with me?
Yes.
And then are the things,
The endomembranes.
What's that?
are those things coming off vesicles?
Yeah, watch.
The membrane system consists of the endoplasmic reticulum and Golgi apparatuses. Vesicles pass from one part of the endomembrane system to another, and secretory vesicles from the Golgi apparatus merge with the plasma membrane, secreting their contents by exocytosis.
So they're showing you, see how these vesicles break off and move? See, they show this vesicle.
Secreting their contents by exocytosis.
This vesicle is going toward the Golgi. This vesicle has broken off the Golgi and is leading outside. See, it's leading outside. And eventually it gets to the outside and spills its contents out. Whenever we're secreting contents from the cell, that's called exocytosis. That's releasing contents from the cell and squirting it out. Isn't that cool?
How come one of them went through and the other one didn't?
I don't know. Well, that one's not there yet. They're trying to show it in 3D, and it hasn't reached the outside yet.
What does it release?
Just whatever's in there. And it depends upon, it could be saliva. It could be snot. It could be digestive enzymes. It could be...
what's it called?
It could be poison, some kind of poison that's being squirted out of the cell. You know, there are Golgi apparatuses in the mouths of rattlesnakes that make poison and secrete it. And so when that rattlesnake bites you, the poison goes into you.
Venom, right?
Venom, yeah. Let's see another one. Ooh, this is a good one, y'all. This really shows the membrane really well. Yeah, this is a good one.
This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane. They leave the cell instead by a process known as exocytosis.
Let me pause it here. Let's all yell out at the same time what things are that I point to. You want to?
Mm-hmm.
Okay. What is this?
Golgi Body.
Try and say it at the same time. A good class says everything together.
Body or
unity. Let's show
apparatus?
some unity here. Ready? 1, 2, 3.
Golgi Apparatus.
Okay. Pretty good. Golgi apparatus or Golgi body.
Okay.
That works.
Endoplasmic Reticulum.
That's endoplasmic reticulum. What about?
Nucleus.
That's the nucleus. What about?
Mitochondria.
That's a mitochondria, and we hadn't learned about that. That's going to be tomorrow. And all of chapter 8. All of chapter 8 is about the mitochondria. Mitochondria, very exciting.
Oh, yeah?
Jessica, you excited about the mitochondria?
She's so excited.
What about
She
this? What's
honestly,
this?
she can't contain herself.
Okay, this is the plasma membrane. Let's say it together on 3 when I point. 1, 2, 3.
Golgi Body.
Golgi, let's say apparatus. You ready? 1, 2, 3.
Golgi Apparatus.
Golgi apparatus. 1, 2, 3.
Golgi Apparatus. Endoplasmic Reticulum. Nucleus. Mitochondria. Plasma.
Plasma membrane. Okay, you ready?
Plasma brain.
Here we go. Watch it.
After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus. Their membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As membranes merge again, the contents leave the cell without actually crossing the plasma membrane.
Ugh.
Oh, that's cool.
Huh?
What does it become?
Wait.
This process can also occur in reverse, allowing large molecules to enter a cell.
That's cool.
This is termed endocytosis.
See, that video is real good because it shows the membrane. Do you see how the let me pause this.
So,
you see how the membrane becomes the vesicle? It's the same stuff. So as it pulls these molecules in, it wraps around them and breaks off, you see? And breaks off, and now we have a vesicle. The vesicle used to be the membrane, the plasma membrane. Yeah?
So the cell membrane is constantly getting like bigger and smaller?
It's constantly getting a little bit bigger and a little bit smaller. As it takes stuff in, it gets a little smaller. And as stuff goes out, it gets a little bigger.
That's how cells, like, is that how infections work?
Well, an infection, these molecules could be foreign bacteria.
Yeah, so they get inside and eat it.
That can get inside the cell. And what a white blood cell will do is capture these things. So a white blood cell will wrap these things up and pull it inside, and then it'll destroy those things when it's inside them. It'll take special. There are special vesicles that contain digestive enzymes, and we call them lysosomes. You see that word lysosome? Lysosome is my favorite organelle. I like to call it a floating ball of death. That's what a lysosome is. It's a floating ball of death. And it will destroy stuff. It's a ball of digestive enzymes that will destroy anything. Watch, this is a video that shows a lysosome destroying something. It's a great video. Watch this one.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus.
So there's a lysosome.
When particles such as viruses or bacteria are ingested by phagocytosis. Okay,
so here is some foreign bacteria coming in. This is maybe you stepped on a nail or something, and there's some foreign bacteria coming in. And this big cell here is called a white blood cell. It's going to kill those things for you. Watch how it does it. It uses these lysosomes that it just made in the Golgi. Watch this.
Bacteria are ingested by phagocytosis. The lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes.
Look, kills them.
Lysosomes also fuse with organelles, such as old mitochondria.
We need to destroy that old mitochondria?
This results in the destruction and recycling of these structures.
Wasn't that great?
Does it get rid of like dead cells?
Yeah, it'll kill dead cells too. Did you know that all of you were born with webbing between your fingers and toes?
What happened?
Wait,
Emery, you getting into this?
Wait, is that why some...
It's something about this area right here.
Nope, astronomy. I was
Is that what it
every
is?
day.
Is that what you slept when you were over there?
Yeah, every day. I could not say wait safely.
Yeah, you can. Listen to this, y'all. We all had webbing between our fingers and toes when we were developing in our mother's womb. And what happened was, the reason why you don't still have the webbing is because it was eaten up by lysosomes. The cells killed themselves. The cells of the webbing were informed to kill themselves. It's called cell suicide.
Really?
Scientifically, it's called apoptosis. And what the cell does is makes a bunch of lysosomes and releases the lysosome on itself. And the lysosome eats up the entire cell, and the cell dies. And that happened to all the webbing between your fingers. Now, in some people, the instructions are screwed up, and they don't break down their webbing, so they're born with some webbing still there. And what a great swimmer you'd be. Yeah? You could be, if somebody just happened to have a mutation where they were born with all the webbing between their fingers and toes, they would win, they would be the fastest swimmer. But it's never happened to anyone as far as I know.
Wait, what is...
been born with all of it.
I know that whenever the cell secretes and releases, it's called exocytosis. What's it called when it comes back in?
It's called exocytosis when it releases something. It's called endocytosis when it takes something in.
Okay.
In particular, if it takes in a large organism like these bacteria, when it takes in bacteria, once you take it in, whoop, that's called phagocytosis. Phagocytosis is taking in big bacteria. Yeah, it's a funny word, phagocytosis. You know what it's called when it takes in small particles like little water droplets? It's called pinocytosis. It was always easy for me to remember phagocytosis and pinocytosis. Phagocytosis is large particles, and pinocytosis is small particles. That's easy stuff to remember, right? They actually changed the name of it because it sounded so dirty. They changed it to phagocytosis and pinocytosis. So people wouldn't say phago and pino. Yeah, you had your hand up.
Okay, if we didn't have lysosomes, would we like die every time we got a paper cut or something?
If you didn't have lysosomes?
Yeah, because it like gets rid of all the foreign stuff. Like, what
Yeah, probably so. You'd need something to get rid of the foreign stuff. Because that bacteria would get in you, and then you wouldn't kill it, and you'd be in a lot of trouble. It would multiply, you'd get an infection, you'd die. Yeah.
So people say like you need white blood cells. You don't actually need white blood cells. You need the lysosomes in the white blood cells.
That's right. Yeah. The white blood cells have a lot of lysosomes and kill stuff for you.
Okay, so red blood cells don't have lysosomes?
No, not much. We'll talk about red blood cells later, but
Okay,
they don't.
so I have another question.
Yeah.
You said when they go out, something happens? How do they leave?
If you release stuff to the outside, if you would release stuff to the outside like that, that's exocytosis. Exocytosis is releasing to the outside. Endocytosis is taking something in.
Oh, so it didn't show exo.
No, this video didn't show exo.
Okay, so you showed it.
The video before showed
So
you.
how do you release bad things? How would you do that?
If you want to get rid of, you got some waste you want to get rid of, you just exocytosis it. You just release it to the outside.
And that's why you
So
need
when you
kidneys
kill
to
someone,
purify
it has
blood.
to be taken out.
Yeah, kidneys do that. What's that?
After you kill someone, it has to be taken out after.
Yeah, that's that.
Yeah, after you kill something and it's broken down, it would probably release the waste contents outside. Unless the cell wanted to use that waste to build something. Then the cell could take that in and build something out of the waste particle.
Like what? Where does it go once it comes in?
Yeah, what is outside? What is
like,
it? Yeah.
Once it releases.
What's outside the cell?
Yeah.
Well, it could just be the body's fluid, like the blood. And then the waste products would then go through the blood, and there's a special organs you have for filtering the blood of any waste particles, the kidneys, and then you end up peeing it out. You pee out all this damage, all this dead stuff, all this dead particles come out in your urine. That's part of what your pee is. All that smelly stuff that comes out, the smelly yellow stuff is part of the waste products that you get rid of. You know they test your pee, if you do a lot of drugs, and they want to see what drugs you've been doing, because drugs are like poisons that have gone through your system and filtered out, and you pee in a cup, and they can test your pee to see if the poisons are in there, to see if the alcohol is in there, or the drugs. They can see what's been in your body.
Or poppy UPC muffins.
Poppy seeds, that's right.
Where does it go once it comes in?
Once it comes in,
Yeah, like if it's not bad.
then it'll actually be recycled. It'll go into the ER, and the ER can use the good stuff and build new things out of it, things that your cell needs.
Protein?
Yeah, protein. There's like this bacteria is made of protein. It's got fats in there. It's got lipids. It's got all sorts of things that the cell can use.
Is it like vitamins? Like you can put vitamins? Is that where vitamins go?
Yeah, anything can go into the ER and be used to build new things. End up in the Golgi and be building new things.
When you have endocytosis and they come back in, does it go through the Golgi body and then the endoplasmic, or does it go around the Golgi body and then in through the...
That's a good question. I'm not really sure. Okay. There's an entire class on that stuff called cell biology that you can take in college that goes into the details of everything that's happening in here, which you don't have to know for this course. You just have to know basically what the Golgi does.
Okay.
It takes stuff in and puts it together and releases it. And so that's all you really have to know for this class. You want to see? We're almost out of time here. Whoops. I'll show you a real cool video of what goes on inside the cell. We won't even have time to watch the whole thing, but it is cool. It's called The Inner Life of a Cell. You can get it on YouTube if you want to watch it tonight.